Welcome, ladies and gentlemen, to ISMB to 2010 in Boston. 13 years of ISCB, the society that runs this meeting. We have now 722 members. The membership is almost doubled over the last 6 years. Thank you for becoming members. Those are elected officers. And you will see posters outside, somehow part of it is you. Thank you for showing it. The mission is to advance the understanding of systems through documentation, to communicate the significance of our science community, the government, and the public at large, to encourage and support the next generation of computational biologists. Again, this is by the students who are pushing this meeting further, and to benefit science and scientists. This year we have the first public lecture, and we have the first time organized by friend Lude centers for high school teachers. So we are reaching out to earlier than before. This year's ISMB is special. We have 3 ladies. The chair, Olga Transkaya, John Michael Lineal. The person who is doing all the work behind is the ISCB conference director, Stephen Lurt. You will see run around. This year, we expect over 1,600 participants at the main ISMB meeting. Overall, there will be about 2,000 people participating in one of the events that will happen here within 6 days. We have about 160 talks, and in 8 different tracks. And I wish you a vibrant meeting. The one thing I forgot, thanks to the bloggers. I feel that you help us a lot, and let's also excuse ourselves. I hope that the network is better than it was yesterday. Some unseen problem from what you may call a higher force. It clearly was a higher force. At least that's what we hope at this point. Enjoy the meeting. And with that, Olga Trojanskaya as the chair of ISMB 20. Thank you.
Hi, everyone, and all of us, my incredible co-chairs, Jill Mesrov and Michal. And on behalf of them and myself, I really wanted to thank you and welcome to Boston, ISMB 2010. We have an incredible scientific 8 tracks covering all sorts of different things, from papers to new research and labor to workshop tracks, specific sessions, all sorts of exciting stuff that I hope you'll enjoy. And I just wanted to get to the keynote, but first I want to thank people without whom this really wouldn't happen. Of course, as my co-chairs and Burkhard, wherever he went, Burkhard, the president of ISCB, our incredible organizing committee, the proceedings track chairs, Mona Singh and Joel Bader, the organizing committee, reviewers and volunteers. Nothing would happen without you. Of course, all of you have submitted papers, or posters, are giving talks, are really key part of this meeting and really the meeting itself. And last but not least are really the people who do all the work to make this happen. And without them, this really wouldn't happen. The conference organizing director, Stephen Lee, I don't see him, I'm sure he's running around making sure this actually all works. B.J. Morrison McKay and Susie Smith, the team that really makes this meeting run and happen, and we really want to thank them all. Thank you guys. Now enjoy the meeting and have fun.
Thanks, Olga. You bring new energy, wonderful energy. One point I missed. This is, we are computational biologists. This is our meeting. This is where we all get together on the life field. It's a truly unique event. Let's celebrate that in some sense. So, is the chair of the awards committee, who has been putting in place a few years, a lot of work to make the awards become part of a good tradition. Søren is an outstanding chair, and I thank you for your speak. Unfortunately, he's going out. This is the last year that he has been chair.
Yes, thank you, Burkhard, and good morning to all of you. Welcome to the first session of ISMB 2010. As the chair of the ISCB Awards Committee, I would like to invite you to this year's Overton Award ceremony and presentation, which is one of the two prestigious prizes awarded by the Society in the general area of computational biology. The Awards Committee is composed of a rotating group of current and past directors of the Society, along with previous award recipients, and it selects honorees for the ISCB Scientist Accomplishment Award that will be given tomorrow, and the ISCB Overton Prize that we hand over today. Overton was established in the year 2000 in the memory of G. Christian Overton, a major contributor to the field of bioinformatics and member of the ISCB Board of Directors, who died unexpectedly earlier that year. Christopher Birch of MIT in Boston was awarded the first annual Overton Prize during ISMB 2001 in Copenhagen, Denmark, and since then the annual prize has been awarded for outstanding accomplishment to a scientist in the early to mid stage of his or her career who has already made a significant contribution to the field of computational biology, either through research, education, service, or a combination of these areas. The awards committee makes the selection by reviewing nominees, and you can find information on how to forward a nomination to the committee on the Society web pages. I will encourage everybody to make nominations for the two awards, but I would also like to state that it is no secret that the competition is very tough. The bioinformatics and computational biology research fields are now very large and quite mature, and the selection process requires lengthy discussion before the final choice is made. It is therefore a considerable honor to receive the awards. The candidate is not only highly regarded by the committee, but in fact received recognition from the entire community working within computational biology. It is with very great pleasure that I invite Stephen Brenner to give this year's Overton presentation. Stephen has had an extremely successful and innovative career covering an impressive range of topics, initially working on de novo design of proteins, then going on to his pioneering work on protein domain classification, then transcript maturation via pre-messenger RNA splicing and nonsense-mediated mRNA decay screening that we will hear about today in his talk, to characterization most lately metagenomics. It is indeed truly impressive that Stephen Brenner has been able to maintain such a broad scope at a very high level of quality for a long period of time. Within metagenomics, it easily contributes to the interpretation of the global ocean survey data collected by Brenner and colleagues using his pioneering methodologies, and he is now working on the molecular underpinnings of Crohn's disease using microbiome data collected from human blood. Human genome sequences vary considerably, and so does our other microbiomic genome, with substantial variation observed between individuals. Work in these new areas will be highly important in the era of personalized medicine. Stephen did his PhD with Cyrus Chocha at the Laboratory of Molecular Biology in Cambridge, UK. There he was one of the key authors of the SCOP database of protein structure classification, which is widely used today. This resource is enormous value to the biological community, providing a classification scheme for the fast-growing body of structural data. Without SCOP and its counterpart CATH, the field of structural bioinformatics would simply not be so established. Stephen Brenner did in fact much more than just develop resources. He undertook important research that leveraged the resources he developed, for example, studying evolution from a structural perspective. Interestingly, the work which now has accumulated more than 3,000 citations, was only published in JMB after a fierce battle between editors and reviewers, a story which has often repeated itself with many similar papers in bioinformatics and systems biology. Stephen moved to the University of California at Berkeley in the year 2000. The following year, he was awarded a prestigious Searle Scholarship to exceptional young faculty members in their first independent position in biomedical sciences and chemistry. He has remained at Berkeley ever since and has been promoted rapidly, with his full professorship announced early this year in 2010. His growing lab now includes experimental biologists as well as bioinformaticians. Most recently, he has become interested in mapping human genetic variation and relating it to disease. He set out his vision for personal genomics in a short paper in Nature in 2007 entitled Common Sense for Our Genomes. And for the last few years, he has been developing that vision in part through his involvement in the Human Variome Project. Stephen Brenner has produced, in fact, a large number of impressive commentaries and reviews, many of which have been published in journals such as Science, Nature Structural Biology, Nature Reviews Genetics, Trends in Genetics, and Current Opinion Structural Biology. Stephen Brenner is obviously a scientist in the mode of sharing, also evidenced by his contributions to the increasingly important infrastructure of our field, for example, the Open Bioinformatics Foundation and the BioPearl project. He has been investing time in community-wide efforts which have been to the benefit of numerous others in the fields of informatics and systems biology. Going through his publication list, and I would encourage you all to do that, I noticed that his broad scope also include a 2004 paper published in the Annals of Improbable Research. It's entitled The Morphology of Steve. And in the paper, the many Steve authors show, among many other things, that individuals break Bergmann's rule, which suggests that warm-blooded animals of the same species will tend to have larger body sizes at higher latitudes. Large-bodied animals have less surface area for body mass and are therefore more heat efficient in cold climates. Birds and mammals generally show agreement with Bergmann's rule, but the paper that Steve has co-authored demonstrates very clearly that larger-bodied Steves do not, in fact, occur more commonly in colder environments. This paper is not among Stephen Brenner's more than 20 papers with more than 100 citations. Still, I would submit that it can be highly recommended, and it indeed is enjoyable reading. So, Stephen, we congratulate you with your career so far and congratulate you with the prize, and we look forward to your presentation, which again confirms that you are an excellent wordsmith. The title that you can see over here is Ultraconserved Nonsense, Gene Regulation by Splicing and RNA Surveillance. So, please.
It is a profound honor to be here today, especially as I think about sharing this podium over the course of the meeting with so many of the people who have mentored and inspired me in my scientific career. And especially having a talk in honor of Chris Overton, who in his foreshortened life did so much to identify and surmount, both scientifically and sociologically, hurdles that still challenge us today. But I think most of all, this talk is a recognition and my collaborators, and above all, my students and postdocs. And it's for them that I actually am pleased to be here. So today I'll be talking to you about nonsense. From Berkeley, normally nonsense comes in the context of politics. And indeed, the title of this talk was once misinterpreted by a colleague, as you might expect at Berkeley. As soon as you mention the word conservative, it's only one way that can be interpreted there. But in fact, the nonsense I'll be talking about is different, literary. What I have here is the first stanza of the poem Perhaps the quintessential nonsense verse, I'll read it to you. "'Twas brillig, in the slithy toes, in the way, all mimsy were the borogoves, no brow." What I'll try to demonstrate to you over the course of this talk is how has been recapitulated, or actually precapitulated. So why is it this confection of portmanteau and joke so widely preserved within English language culture? And in fact, original publication through the Looking Glass in 1871, but in fact, a newsletter for that, where it was first published, in a journal he called Mishmash, I'd like to celebrate it into the novel. Preservation goes beyond just simply even the bizarre words that it comprises, but goes as far as the details of pronunciation. Those who want to know exactly how to say the words and would have deep arguments about this, and Lewis Carroll felt obligated to respond to them as he would publish later versions in future. So why has it been so well preserved? Well, there are literally books on the topic, and I shall not address all the reasons, but as one looks at it and listens to it, it retains its sentence structure. While the words have no meaning in English, one has a sense that they are implying. You understand where the nouns, the verbs, the adverbs. And beyond that has this beautiful rhymed iambic tetrameter, which gives it an underlying sense and meaning as well. So I'm going to argue that this underlying meaning where there is nonsense, is what we see in biology. Now, of course, normally nonsense is bad, whether in politics or in language or in referee calls in football. And of course in biology, where it can be fatal. And in nonsense biology refers to nonsense codons, most often thought of as being mutations, where you have a normal codon which codes for an amino acid, and then you have a mutation which creates a nonsense codon, which would lead to a truncated protein were that gene to be transcribed and translated. And the problem with nonsense mutations is that in many cases they lead to a dominant negative phenotype, as I'm going to illustrate here in this cartoon. Imagine the protein here. This protein here is a transcription factor with a DNA binding domain and a polymerase recruitment domain. And so its operation normally is to have each one of the two alleles bind to a given binding site and drive transcription from a site proximate to it. If you end up having an entire allele deleted in the hemizygote, if this gene is properly regulated, then one of these will still be able to bind to that site, and you will still drive transcription. Potentially at a lower level because you have less protein around, but still one allele will be present to be able to lead to some proper transcription occurring. But with a truncated protein, the consequence of the nonsense mutation, that truncated protein will some of the time bind to that binding site in the DNA, and it will not drive the transcription, but furthermore, by occluding that binding site, will prevent the other complete allele from being able to bind there, and as such, it will end up turning the gene off and have the dominant negative effect. Indeed, because this one will be smaller, then tropically it'll actually be favorable for this truncated one to bind there. And so because it looks like many mutations will be dominant and negative, studies in worms with truncated mutations would have a dominant negative phenotype if not ameliorated. It's thought to be this is the motivation for having a system in cells called nonsense-mediated mRNA decay. This is an RNA surveillance system which recognizes mRNAs with these early stop codons and causes them to be degraded rather than being translated to make protein. And the consequence of this is it converts the truncation state here into the hemizygote, or haplosufficiency, and is able to act in the same way as the hemizygote. And thus being able to remove these forms which would be deleterious, we're able to, in many cases, transform a disease which would be dominant to which will be less severe or entirely recessive. And it turns out that what I showed you in that last cartoon was not just simply a cartoon, but an example of what truly happens with a gene called SOX10. SOX10 is a transcription factor. They were mapping out the relationships between mutations in it and the phenotypes observed. Really serious. Mutations which early on led to a number of phenotypes which are certainly undesirable. Warrensburg syndrome and Hirschsprung's disease, which lead to strange pigmentation, some very minor neurological defects, and can lead to also bowel obstruction. Not desirable characteristics, but ones which lead individuals to live normal, healthy, happy lives by and large. But mutations which were near the end of the gene led to a much more severe set of complexes, including peripheral neuropathy and peripheral lipodystrophy. So how is it the mutations that were early were mild, and that mutations that were early were mild, whereas truncated mutations which removed much less of the protein have a much more severe effect? The answer here, again, relates to the action of nonsense-mediated decay, NMD, this RNA surveillance system, in that the mutations that were early were recognized by that system. And therefore, the truncated RNA was cleared from cells, and those truncated proteins were not produced. There was less protein to these more minor syndromes. When the mutations were very near the end of the gene, though, NMD wasn't able to recognize that these were premature. It led to a very slightly truncated protein being produced. But slight truncation was enough to lead to a much more severe set of complexes. And it's been later shown that, in fact, if you do nonsense-mediated decay, you're able to increase the amounts of these proteins that are produced, and show that nonsense-mediated decay is almost certainly responsible for these mutations having a less severe phenotype. Nonsense-mediated decay has a number of other roles within the cell beyond recognizing these truncating mutations and clearing them. It's actually essential for normal development, especially in the role of maturation of the immune system, because VGA recombination is imprecise, and nonsense-mediated decay is thought to be responsible for clearing many of those imprecise recombined forms, which would otherwise be deleterious. It turns out that the processivity of polymerase is also such that a significant fraction of normal mRNAs will actually have early stop codons introduced into them, and nonsense-mediated decay is responsible for moving those from the pool and preventing those from making protein as well. The topic I'll be discussing with you primarily is in the field of splicing. That splicing can happen in several ways of splicing. One is that if you missplice into an intron, most often that will lead to an isoform which is an early stop codon. These two will be recognized by the nonsense-mediated decay machinery and cleared by the cell. So I want to give a thought for a moment to how it is the cell is able to recognize an mRNA which has an early stop codon and lead it to be degraded, and distinguish this from a normal transcript with a normal stop codon and lead that one to be translated rather than being degraded. But it turns out, actually, this is a mystery for us in species. That outside of the eutherian mammals, we do not know how it is that nonsense-mediated decay works. My own lab has done work studying nonsense-mediated decay in the fly. Other groups of the worm. In both cases, we know every normal mRNA that is degraded, differentiated from the cell, from the mRNAs that are not. Nonetheless, we do not know what rule the cell uses to distinguish those with early stop codons that are degraded from those which are not. But in mammals, there's a rule which seems to describe a large fraction of all the targets of nonsense-mediated decay. So this rule of thumb is one which I'll be using beyond there. And this rule of thumb is based on the process by which mRNAs are actually produced in molecular detail. As mRNAs are produced, you splice out introns, but importantly, as those introns are spliced out, there's a complex of proteins known as the exon junction complex, which is deposited short of each exon-exon junction. These exon junction complexes were first identified as important for nuclear export. In fact, it's thought in some cases that the reason that mRNAs get exported and introns do not is the mRNAs harbor these exon junction complexes. Indeed, there's evidence that single exon genes need special adapter proteins to promote their export because those mRNAs are lacking these exon junction complexes. But it turns out these exon junction complexes play another role, which is critical for RNA surveillance, that they act as a memory for where those exon-exon junctions have taken place, where the neurons had been. And the model is that the polymerase, as it scans along the transcript in its first pioneer round translation, will displace all those exon junction complexes until it reaches the stop codon. Once it reaches that stop codon, there will be exchange of factors at the GCAP, stabilization of the transcript, and then typically the transcript will then undergo several thousand rounds of translation subsequently. But if you have an early stop codon, and it turns out that an early stop codon in the case of human and mouse is defined as a stop codon that's upstream of this last exon-exon junction by more than about 50 nucleotides. As the ribosome scans along during the pioneer round of translation, displacing each of the exon junction complexes as it goes, when it reaches that stop codon, you can see there will still be an exon junction complex remaining. Between release factors and members of the exon junction complex will end up leading to nicking, deadenylation, or decapping of the transcript, and ultimately its decay. And so this is a simple way to be able to look at a transcript and be able to determine whether it's likely to be translated normally, or whether it's likely to be degraded by this nonsense-mediated decay machinery. In that if your stop codon is on the last exon, or approximate to the last exon, it will be a normal stop codon, it will be translated. If it's more than 50 nucleotides upstream of this last exon-exon junction, then it will most likely be degraded by the nonsense-mediated decay machinery. Now, I told you this is a rule of thumb. It does not apply to every transcript in the cell, but it's one which tends to be generally applicable to most transcripts within human and mouse cells. The nonsense-mediated decay machinery is found in all eukaryotic species that have been studied, and it does involve the same set of key proteins in all species that have been studied. Proteins called UPF1, UPF2, UPF3 are the core proteins, with other proteins called SMUGs that elaborate it. But the mechanism by which they recognize outside mammals is different. So now I'm going to change gears and talk a little about splicing it. So splicing first came into public view with the complete sequencing of the entire human genome. After the genome was sequenced in its preliminary form, we got wonderful titles on the front page of the New York Times, like this one here that I love, that we survive on a low number of genes. Incredible, isn't it? And furthermore, that our pride was truly in for lows, as we had barely more genes than the lowly flying worm, and wasn't this horrible. And we felt compelled to come up with explanations for this. And remarkably, as reported in the New York Times, no less, 2 days later, we knew the answer. The answer was that we didn't have more genes, but we had better genes, because our genes had more alternative splicing, and alternative splicing was the answer. We had more proteins, and basically demolishing the old view of one gene, one protein, we had more proteins, and this is why we were better than. And then there were wonderful papers, such as this one by Pierre Bourque, which tracked the amount of alternative splicing that was recognized in human genes. So basically, this is the percent of human genes that have alternative splicing. This is looking at years, and you can see that back in 1994, we thought a handful of genes were alternatively spliced based on studies that have been done. And then extrapolate this out, and Chris Burge and others did work a couple of years ago showing that indeed virtually all multi-exon genes do indeed exhibit alternative splicing. But this extrapolation taken out to 2010 gives about 135% of all genes exhibiting alternative splicing. I used to think that was a great joke line, but in fact, it may have been that the work was prescient. If we look at all the large number of non-coding RNAs that are now being discovered, and many of which undergo splicing, 135% of the protein coding genes may not be a bad number. But one thing that I want you to recognize as you think about this, alternative splicing can be a mechanism for introducing premature termination codons. Certainly missplicing can lead to introduction of premature termination codons, but also you can have what I call poison cassette exon. An exon like this one here, which would incorporate into the mature mRNA, will end up introducing a stop codon. And so one isoform will produce protein, and we term this productive splicing because it produces protein. And one form will have this poison cassette exon, which will poison the transcript, leading with this premature stop codon, leading this one to be degraded by nonsense-mediated decay. And we term that unproductive splicing because it leads to an mRNA that will be degraded by the NMD machinery and will not be translated to make protein. So several years ago, work in my lab discovered that this was something that was surprisingly common. That a majority of all the different mRNAs we were looking at that we could identify at that time alternative splicing actually had at least one isoform which could be targeted for nonsense-mediated decay. And it was a significant fraction of all the alternative isoforms looked like they would be targeted for nonsense-mediated decay. This was profoundly disturbing. In fact, we did not believe the result the first time we found it. Another way, and then finally became convinced it was true. However, the first three sets of reviewers we sent it to didn't believe it was true. The fourth one did. Because it made no sense. It was like taking something and then immediately putting it into a shredder. And I looked for years for an image of this, and actually last year you could buy one of these from ThinkGeek, the Shrinter, which is 4-toner dry type electric figure hard steel covers. You can only buy that on April 1st of that year. But why is it that our cells are producing all these mRNAs whose only fate is to be degraded? Well, there's some work suggesting that many of these are just noise. They're not under selection. And the cell produces lots of mRNA all the time that's just being degraded. We call it introns. And so maybe the cell just produces more mRNAs that don't make anything, and they get degraded as well, and the cell just doesn't care. But there was one example that had been discovered shortly before we made this observation about the pervasiveness of these mRNAs targeted by nonsense-mediated decay, where actually was a mode of gene regulation, a gene called SC35, which auto-regulated itself. I'm going to show you a cartoon which shows how you can do regulation via nonsense-mediated decay. I'm showing you here are two nuclei which are exactly identical in terms of their genome, in terms of the pre-mRNA being transcribed from that genome, and in terms of the mRNAs that are being produced, except for one detail. And that one detail that's different is based upon the fact they have slightly different splicing factors in them. And the red splicing factor that's here in the nucleus on the right, but not present in the nucleus that's on the left, will act analogously to a transcriptional oppressor. That red splicing factor will have the effect of turning off the translation of this gene. Because that red splice factor leads to the inclusion of this poison cassette exon, and then as these mRNAs get exported from the nucleus and undergo their pioneer round translation, the ones on the left will end up being translated normally, whereas the ones on the right will be degraded by nonsense-mediated decay machinery, and so it will not produce protein. Decay, coupled with alternative splicing, can be a mode of gene regulation. In the case of SC35, it uses a mechanism analogous to the one I'm showing you here, where in fact, as you get more protein, it binds back to its own pre-mRNA and leads to production of an unproductive transcript which will not produce protein, and so you are able to have homeostatic control the level of this SC35 protein in order to maintain appropriate levels within the cell. Now, of course, SC35 is not simply acting to go and make this nice loop dancing with itself, but rather it's regulating the splicing of many, many other transcripts in the cell. And indeed, there were hints in the literature that maybe this could be a more pervasive feature of splice regulators, in particular from the main family of positive regulators from splicing and tissue splicing known as SR proteins. So here's the family portrait of SR proteins. There are 11 of them in human, of which 10 are conserved with mouse. They all come from one ancestral gene, and their gene duplications, which lead to the current family. All of them have one or two RM domains in this RS region, and this reflects actual rate. They bind in a sequence-like manner to RNA through these RM domains, and this RS is responsible for promoting movement of the spliceosome to that region. So the mechanism by which they operate is relatively straightforward. And so what we discovered was, in fact, in one of these SR proteins, that there was alternative splicing which introduced a premature stop codon. Here's a gene called SRP55, one of the members of the SR family of splice regulators. We discovered this alternative exon here, which when incorporated, we predicted would be degraded by the nonsense-mediated decay machinery. And in fact, we didn't find it just in SR55, but indeed we found this in every single member of this family. Now, we were looking very carefully to try to identify these, so we didn't know when we first found this whether, in fact, this represented the fact that this family was special, or whether it reflected the fact that if you simply look in any family carefully enough, that there's enough missplicing going on that you'll end up finding mRNAs which will be targeted for nonsense-mediated decay. So we had no idea whether, in fact, this was something which is really interesting and important, or whether it was just simply noise. In fact, we couldn't really be sure all these were truly being degraded by nonsense-mediated decay, so it was actually to assess this that I opened the experimental part of my laboratory, where we inhibited the nonsense-mediated decay machinery using knockdowns to that key effector of nonsense-mediated decay called UPF1. What we'd expect to see, if indeed this was being degraded by NMD, was an increase in this. We used quantitative real-time PCR to measure this, and in fact what we found was a vast increase in the amount of the mRNA degraded, which with the early stop codon, indicating that it was indeed being degraded by nonsense-mediated decay machinery. We found that, in fact, that while the targets of nonsense-mediated decay, according to the databases we were looking at, appear to be very minor isoforms, typically comprising about 5% of the cellular mRNA for this gene, that in fact, actually, it was the major isoform produced by the cell, but NMD was simply clearing most of it and leaving just a tiny bit remaining that we were able to identify. The fact that this was such a major component of the mRNA that was being produced by the cell, and the fact these numbers are often fairly close to 50-50, which is something which requires typically regulation, suggested to us, or at least hinted to us, that this may be something that was actually important and not just simply noise. And indeed, we found that this degradation by nonsense-mediated decay was experimentally verified in all the members of the family. But how do we know whether it's really, truly, functionally important, other than making teleological stories? We use conservation. That Chris Lee had observed that most isoforms, minor isoforms, are not shared between human and mouse. So if you do find a minor isoform shared between human and mouse, that's highly suggestive of a functional role for that isoform. As you can see here for SRP55, not only is the minor isoform, including splicing exon, conserved, but in fact, it's very precisely conserved with the exact same boundaries of those exons there. And indeed, in all the cases of these which were present in mouse, we found the exon or the alternative splicing, which otherwise created nonsense-mediated decay, was conserved with mouse. This told us that almost certainly these were here for a function. So what could the function be? Well, a group in Italy has unpublished work showing that, in fact, it has an important role in preventing cancer. That indeed, there's a cancer which was known to we have splicing this raw oncogene, which creates a form which leads to more aggressive cancers, that it was downstream of the RAS-ERK pathway. And they found the key thing linking these was actually a change in the unproductive splicing of a splice factor ASF-SF2, one of the proteins I showed you on the previous slide. More generally, we suppose this is actually just a mode of regulation of these genes. What was known before we began this work was that SC35, the splice factor, was auto-regulating itself. When you had more SC35, it would block production of further SC35. We and others have now demonstrated this is true for a protein called ASF-SF2, which we suspect, but have not yet shown, it may be true for other members of the family as well. I'm actually going to skip over this experimental data and tell you that we think that going beyond that, that's not just simply auto-regulation of these, but in fact the SR genes can in many cases compensate for each other. While different levels of SR genes are responsible for tissue-specific alternative splicing, that sometimes if you deplete one SR protein, you can then recover normal activity by increasing the abundance of other SR proteins. And what we believe may be happening is, in fact, that these SR proteins are all talking to each other via the coupling alternative splicing and nonsense-mediated decay. And we've actually demonstrated that ASF-SF2 is talking to several other proteins and basically inducing their unproductive splicing when you change the abundance of ASF-SF2. And we hypothesize, but have not yet shown, this may be a complete network where all the SR genes are talking to each other. So as we looked at the SR genes more carefully, and we're examining this conservation, trying to see exactly how these boundaries between these poison set exons were aligned, we did a shocking observation that as we looked at these genes, the most highly conserved region of these genes is associated with the unproductive splicing with that poison cassette exon. Basically covering almost all that exon and extending a little bit upstream of it was a region of 100% identity. This was not simply the most conserved region of that gene, but in fact this is amongst one of the most conserved regions of the entire human and mouse genomes. That it fits into a category identified by Bejarano and colleagues known as ultraconserved regions, which were arbitrarily characterized as being 200 or more nucleotides of perfect 100% identity between human, mouse, and rat. There are about 500 of these in the genome. About 111 of them are within genes. The other ones are intergenic. And here was one of them sitting flying at us in this gene SRP55 and associated with this unproductive splicing. And indeed, this wasn't an outlier. There were 4 more genes which had ultraconserved regions, 9G8, 228 nucleotides. SRP20 had 618 nucleotides of perfect identity between human, mouse, and rat. We don't know of anything in biology which actually demands that much identity. There's something spectacular going on. And for the other members of the family which didn't make the threshold of having the 200 nucleotides of identity, still had exceptional identity. In this case, for example, SRP75, 3 regions for these 3 coordinately spliced exons lead to unproductive splicing with 114, 77, 119 nucleotides. So what is going on? Why are these sequences so conserved? Well, the first place we normally look for conservation in the genome is for association with protein coding. But of course, these aren't protein coding. That we know that they're there to actually make mRNAs that are being degraded. Even if they weren't being degraded, most of the mRNA, most of that exon would be after the stop codon, and so it wouldn't be translated anyway. And even if frame shift past that stop codon, there were other stop codons after it. So it's not making protein. Bejarano and colleagues looked to see whether there were low energy structures by doing folding. And there wasn't any obvious story there. It doesn't mean that there isn't RNA structure. It means that by using the physical methods that were applied, you weren't able to identify it. The better way to look at this would be to look for covariation. The problem is that these ultraconserved regions are ultraconserved, which means there's no variation, which means no covariation. And as I'll show you in a moment, we're doing some work now which allows us to look out beyond human, mouse, and rat, and perhaps be able to see whether there is covariation at a greater distance. The other thing we look for was whether there were overrepresentation of known binding sites, whether they be splice regulatory sequences, microRNAs, what have you. We didn't find any overrepresentation, but I think this also probably reflects the limits of our methods. Because in fact, we found lots of sites, just no more than you'd find in a random piece of DNA. Other groups have looked at some of these in detail using biochemistry and found that for one of these SC35, that there are in fact binding sites for many SR genes. So it may well be that there are many sites present there. Now, the ultraconserved elements that were intergenic have been studied more extensively than those which are intragenic. Several of these have been identified as being transcriptional enhancers, and some of them seem to have a history as being repetitive elements. So we wonder whether these might also be repetitive elements. And what we found was they were not. In fact, they were not similar to any region of the genome. I'm going to come back to that in a moment. because there was no similarity anywhere else in the genome except in recently retrotransposed pseudogenes. Now, the fact there was no similarity between any of these ultra-conserved regions and anywhere else in the genome has a profound implication. Because we'd assumed that given that this unproductive splicing is such a strange thing, in my view, to be occurring, that we found in every member of this family, the parsimonious explanation was that it happened once in the family at the base of the family, and it was then inherited by all the members of the family. But that appears not to be the case. Because in fact, the unproductive splicing involves different splice events in different parts of the genes, and there's no sequence similarity between all these different ultra-conserved regions. So it looks as if, in fact, this bizarre mode of gene regulation has evolved over and over and over again within this family. And we're trying to understand how conservation is strange mode of regulation, yet it reappears. To do this, we looked at the most closely related small clade of the SR protein, these ones here, protein SRP40, SRP75, SRP55. The story I'm going to tell you now is one that's newer work. We don't know all the details yet. It's a little bit less clear. I'm going to try to pull out some of the highlights for you. So here's those two most closely related genes, SRP55, and again, I put the mouse one here, the human one here. Here's the ultra-conserved region overlapping that poison-sted exon going slightly upstream of it. And I'm showing you an alignment with SRP75, which is much, much longer. Here they are roughly to scale. You can see this first intron here, which is shown here, not to scale, is much longer, but there's a very beautiful match between all the coding regions, and the coding regions are almost exactly the same. We do in humans see some very weak evidence of splicing of that same poison cassette exon. But there's much more evidence for the splicing of these other set of coordinately spliced exons, which are probably the main poison exons used by this gene. And indeed, as we look for conservation, it is these ones that are highly conserved, and there's no evidence of conservation here. This may be a legacy, something that's not under selection. These are the ones that are clearly under selection. So you see how it's ultra-conserved, but yet it is somehow changed. I tried to map this out in detail by looking at a detailed phylogenetic history of all the chordate SRP40s, 55 and 75. We looked as far as the chordates because we knew that certainly outside of the chordates that the mode of NMD was different, and so we wouldn't expect to see the same type of regulation occurring. It was very difficult to ascertain actually what the ancestral state was because these guys had very poorly built trees. We had very poor bootstrap values here. And to be able to understand actually which one was likely to be ancestral, most of them looking at the exon intron gene structure. What I'm showing you on here is an alignment of the protein genes, but what I've done is colored each exon differently across the gene. And what you see is that there's very clear conservation of exon structure here, SRP4. Conservation exon structure, these are clearly SRP55. SRP55 and 75, exactly the same gene structure, other than those poison cassettes here. The black lines indicate where it is that we see evidence of the and so S75, as I showed before, has the poison exon here, and 55 in human, we see it just here. From this, we'll actually ascertain the more ancestral versions of SRP40 actually ancestral form of this gene, and then it duplicated to then make SRP55 and 75 SRP75. So this is the big picture. So here, this is new work, and so while you've been welcome to live blog the entire talk up to this point, the few slides which have this little symbol here are indications that while you're welcome to share this with your friends, take pictures of it and so on, please do not make this available in some archival form. Shows basically the history we've been able to ascertain so far. We actually found, to our amazement, conservation of the unproductive splicing based on the EST and similar data, not all the way out from amongst the chordates, but in fact out into the full set of animals, and indeed all the way out into the fungi. Meaning that the entire episticont lineage, the clade that joins the fungus in animals, appears to have had unproductive splicing in its ancestor. Indeed, we have done recent experiments to prove, indeed, that in Neurospora, that there's a retained intron in the exact same intron where we see the unproductive splicing in human. To our knowledge, this is the most ancient regulated splicing event that's ever been seen, and its role is to cause the transcript to be degraded. Suggests that nonsense-mediated decay as a mode of gene regulation may have been the ancestral reason for having alternative splicing. And its role in protein elaboration may be a recent discovery. And it tells us that the history of unproductive splicing goes back at least 1 years. The date at which this diversion occurred is unknown. It's normally placed between 1.2 and 2 billion years ago. this mode of regulation could be 2 billion years old. And one thing that was most surprising to us as we looked at this was, as I told you, the mode by which NMD works changed outside of this clade here. We don't know exactly when it changed, but we know it was different over here. So it means that even as the NMD mechanism changed, regulation of this specific splice site was preserved. Sort of like laying down the highway at 60 miles an hour and changing your tires as you go. We understand actually what the details of the ultraconservation look like, not just with amongst we're looking for long regions of conservation, but also trying to understand in detail how our goes. So what I'm showing you here is SRP40 in a region just of the exon, and then for comparison, a region upstream of the exon downstream. Highly conserved genes, and so there's a much higher level of conservation in these genes. What you see is that with no gaps introduced in the region that's upstream, you get beautiful alignment all the way out amongst all the vertebrates, all the way out to the fish. And more typical, you see very, very little conservation out there. You see some patterns being tracked and associated with splicing, but generally much less conservation. As we go into the actual exon, the poison exon, the conservation is quite deep. That it's different amongst the tetrapods and amongst the fish. The fish conserve this block, and the tetrapods conserve this block, indicate that there was their common ancestor, and then they diverged into different sequences. And one of the shocking things was trying to understand how it is that SRP 55 and 75 got their new and different highly conserved regions associated with this unproductive splicing. And what we found was actually we could never see any case where we saw both the SRP40 splicing and the SRP55 splicing. What appears is that F5 ended up being duplicated at the base of the chordates, that very rapidly after duplication, it lost the SRP40 mode of regulation and gained this new SRP55 mode of regulation using a poison-cassette exon. So somehow a sequence which we know was being highly conserved was lost. A new sequence, immediately highly conserved, was gained in an evolutionary wink of an eye. And so with that, I'll conclude the things I have told you about in this story. A function for unproductive splicing, a reason why we may actually have nonsense in biology. A role for these ultraconserved regions. We don't know in detail why they are ultraconserved, but we see their clear association with this unproductive splicing, apparently as a mode of gene regulation. And then finally, this incredible evolutionary story, the repeated independent evolution of this mode of gene regulation, one which is extremely ancient, as I said before, the most ancient regulated splice event that we know of, and yet one that's been able to reoccur often, but having re-evolved ends up being exquisitely conserved at the level of sequence. And so I want to tell you finally where it leaves us, back to Alice in the Looking Glass. Somehow it seems to fill my head with ideas, only I don't exactly know what they are. Some of the people involved in this project were Ed Green, who first initiated this project within my lab. The work I showed you today was the work of Liana Leroux, Dan Liang, Orna Mizrahi Man. Gang Wei is a new member of my lab working on this project who's been involved. We have a number of collaborations which have been critical for this. And then I'm going to tell you a little bit more about Maki Inada, a postdoc in my lab. She's told me I can tell you a little more about her both scientifically and also personally. It doesn't relate exactly to the work in my lab, but it's one which has, I think, been very meaningful and had something to do with the directions that I've chosen my lab going forward. So Maki came to my lab having been a visiting assistant professor at Macalester College, and she wrote to me at the time of application that she actually wanted to maintain being a professor at a small liberal arts college working graduates, but before doing so, she wanted to actually get more lab experience, and she actually did all the experiments that I showed you today. After being in my lab for a few years, she applied for positions and actually was offered many wonderful positions. Unfortunately, none of them were proximate to the great position that her husband got at Cornell. And so she went off to Cornell as a research associate working in her husband's lab. She was hoping to go to Ithaca College, but there were no positions open that year. Slightly after she left my lab, several months after she left my lab, she sent us an email which was truly shocking. And what that email said in this picture, which she shared with us later, a CT scan was taken of her. She had come down with a persistent cough. They didn't know what the base of it was. Eventually, her husband forced her to see the doctor. You don't need to be an oncologist, a radiation oncologist, to recognize this is not good. This is a 7-centimeter mass, stage 4 non-small cell lung carcinoma, 350,000 cubic millimeters. Recent work that's been done by Sylvia Plavridis says that if you have a tumor of this size, the cure rate is indistinguishable from 0. So we did everything we could to try to support her. One of her friends made up these bands and showed them all these different ways to try to support her as she underwent her chemotherapy. But really the thing that we were hoping for was this. It's a molecule called Tarceva, and I'll abbreviate the story for why we were hoping that that this molecule might actually do something special for her. But briefly what it has to do is that while normally you would be told that Tarceva would give you very little hope in this situation, if I go to the Tarceva webpage, it tells you basically it won't work, you shouldn't use it in this situation for first-line therapy. But she was given as a first-line therapy. And the reason why it was inferred that she probably had a mutation and later confirmed that she had a mutation, this one right here, in a person called EGFR, which makes EGFR constitutively active. And it's this constitutively active EGFR which leads to the cancer activity. It turns out that Tarceva is a specific inhibitor of EGFR. Most EGFR-based cancers are based on having huge numbers of EGFR. You have to basically hit all of those different EGFR molecules. I'm simplifying, but it turns out that in her cancer, most likely she just had one EGFR gene, but it was mutated, and as it happens, Tarceva binds especially well to that constitutively active mutated EGFR protein. After 3 months of chemotherapy and Tarceva, her tumor shrunk down to 0.5% of its original size. You can almost barely see it here, making her a candidate for surgery. She had surgery. She was deemed cancer-free. She was able to then, even after having a chunk of her lung taken out, be able to return to her favorite hobby, which is underwater hockey. She got a faculty position at Ithaca College, the only one to have opened up in about the last 15 years. She got that. And then this picture, which was taken after having been cancer-free for a little over a year, you can find in the current issue of Fitness magazine. Go out to the newsstands today. It's a July issue saying describing how healthy she is today. You can see that this issue is a very copy of useful information in your scientific and personal endeavors. And then most recently, about a month ago, she actually got her first grant, which will actually be involved in continuing this research. So she was given this drug based on her genotype. And that's the direction that the work I'll be going in my own lab in the future. I'll be talking about tomorrow. Now, this last slide, I want to show you something which I think underscores what it was possible to discover that this would be the right drug for her. And indeed, something which underpins why all of us are here today as computational biologists. We have been the beneficiaries of these twin exponential growths of biological information and of computational power. Something unprecedented in human history. I think it's truly a privilege for me, and I think for all of us, to be able to be the beneficiary of this. And as I've told you so far in this talk, at the outset, I discussed a life that was foreshortened. I concluded with a life that was extended. Both these are lives, both their science and personally, inspired me. I hope that perhaps they may be inspirations to you as we go forward in trying to make use of this amazing amount of knowledge and power we have to make discoveries, to understand life, and to extend lives. Thank you very much for your attention.
Thank you very much for a question. While you may come back for one question, I wonder what happens in terms of when some of the essential NMD proteins are who reviews the reviewers. That we don't know in humans, we don't know individual humans who have ND knockdown phenotype. When it's been attempted to make knockdown mice with ND knockdown, it turns out that they are lethal embryonically, actually prior to implant. And so it's a very, very early lethal phenotype. They're disability in the NMD in individuals and actually perhaps between different tissues. This is an important therapies where it's believed that in some cases that the disease is actually caused by NMD. That seems to be the case in some cystic fibrosis, also in some of muscular
muscular dystrophy. In those cases that people have been giving trying therapies to some extent undermine the activity of NMD. The simplest that develops cause read-through of all stop codons. As you can imagine, while it does like make the protein you want to make them being degraded, it has the effect of not basically causing read-through of all other transcriptions, and this is like deleterious. There's a company, PTC Therapeutics, which is trying to develop specific therapeutics that affect just the early stop codons. But basically, if you get rid of NMD entirely, it will be lethal within humans. In flying worm, the effect is much less dramatic.
Okay, thank you very much. So any questions out there? If not, I think we proceed.
There might be one question.
There's one. Okay, thanks.
Do you want to go to a microphone?
Yeah, I think it's, maybe we take this question first and then you go to the microphone. I think we're 1,600 people here, so I think we need to use the microphones.
Actually, I think they're very similar questions. This is speculative, but I was going to ask you, you told us a question about, or you told us a story about one family of genes, but I was interested in your speculation about how common you think this form of regulation is.
So this form of regulation has been shown by many areas to also be prevalent with HNRMPs. He actually doesn't demonstrate that's truly regulation, but that the same types of mechanisms are present there. HNRMPs are thought as the primary family of antagonistic oppressive splice regulators, whereas SRs are the more positive regulators. And so certainly within factors, it seems to be pervasive. It appears to be less prevalent within other genes. We don't know how prevalent it is, and that's something that we're exploring now.
Yes, over there.
Is this on? Oh,
yep.
There we go. Okay. So right at the beginning of your talk, when you were talking about the way that you laid out the detection system, does that mean that it's totally insensitive to stop codons in the last exon? Did you see any sensitivity to that at all?
The model laid out by Limiquad, known as the nucleotide rule, is that stop codons in the last exon will never be recognized by the nonsense-mediated decay machinery. And if you look at normal human genes, you find almost none of them which have 3' UTR intron, because that would lead to that stop codon being recognized premature, whereas 5' UTR intron are very common.
Oh, okay. So there's pre-selection for that particular mutation?
There's presumably selection to not have a stop codon that will be on an exon other than the last one. But stop codons on the last exon will not be recognized by NMD machinery in human according to the 50 nucleotide model. Again, not the model that's the case in other species, where in fact last exon stop codons will also be recognized for degradation via that machinery. It also means that single exon genes will never be targeted by decay, which has some interesting implications when one considers the families of single exon genes.
So are you able to say at this point whether this is the sort of primary way of regulating these splicing cofactors, or is it just one of many?
I don't think that I can say clearly this is the primary way or one of several. It's certainly the case in the instance of ASF, SF2, studied most extensively, that it actually does have other modes of regulating, transcriptional, splicing, and translational that govern what level expression is. But even then, it seems that this is one of the major modes by which it is regulated. We suspect that for outside the splicing factors, that's more likely to have a tweaking effect. One of the benefits of regulating this way is you can also regulate more rapidly.
Okay, so let us thank excellent presentation. And now we end